Welcome to AP Biology 3.5 Photosynthesis. This is Dr. Joseph. In this chapter, which is part of cellular energetics, we are going to study the energy cycle, about redox reactions, and the energy molecules that come out of these reactions. And we are going to study photosynthesis and its two stages, which are the light dependent reaction and the light independent reaction, also called as the Calvin cycle. And then we will look at different types of photosynthesis strategies: C3 plants, C4, and CAM plants. So everything in life is about the cycle of energy. All energy on this planet comes from the sun. The energy of sun travels that 93 million kilometers and comes to earth, where it hits the plant, the leaf, which is where the energy of the sunlight is captured by molecules called pigments, and that kickstarts a process called photosynthesis, wherein the energy of the sun and the water that is absorbed by the roots of the plants and the carbon dioxide that is in the atmosphere that is taken in through the plant leaves together are used to create carbohydrates. And these carbohydrates become the source of energy for animals and plants, and that energy is released through a process called cellular respiration. And in cellular respiration, we end up creating these high energy molecules called ATP, or adenosine triphosphate. And adenosine triphosphate is a ready to use molecule, very easily accessible across all cellular reactions. And in the process of cellular respiration, carbon dioxide is created, and cellular respiration requires oxygen, which is a byproduct of photosynthesis. So in all, it becomes a self-sustaining system of oxygen being produced by the plants during photosynthesis and carbon dioxide being released by animals and plants during cellular respiration. So we'll look at this in a little bit more detail. So the process of photosynthesis, as we saw, needs the energy of sunlight. Sun's energy is held in the form of pockets of energy called photons. And when photons hit the plant leaves, it depends upon the water that it has absorbed through the roots and the carbon dioxide that it has taken in from the atmosphere. And in two different steps, which we are going to look at, it releases oxygen as a byproduct and converts the carbon dioxide, fixes the carbon dioxide in the form of sugars. So the equation of photosynthesis is carbon dioxide plus water plus the energy of sun, giving rise to carbohydrate plus oxygen. And the equation of cellular respiration is the exact opposite of the photosynthesis reaction, where we are taking glucose and burning it with oxygen to provide energy alongside water and carbon dioxide. So ultimately, it is all about energy. The energy of most of organisms on planet come from sunlight. And sunlight, the magic of life has happened where the energy of sunlight is created or converted into a form that is utilizable, accessible to living things. So in the photosynthesis reaction, there are two steps. One is called the light dependent reaction, and in that reaction, two high energy molecules called ATP and NADPH are created. ATP, or adenosine triphosphate, holds energy in the phosphate bonds. So we've seen the structure of ATP before, and we will take a quick look at this again. ATP has two high energy phosphate bonds, bonds between phosphate molecules. And that energy can be broken down. NADPH also stores energy, and both NADPH and ATP are used to power the next step of the photosynthesis reaction, which is called as the Calvin cycle. In this stage, the energy that is contained in the bonds of NADPH and ATP, which originally came from the sunlight, are used to are transferred. That energy in ATP and NADPH is transferred to chemical bonds, to form chemical bonds between carbon and hydrogen in carbohydrates, so that we can use carbohydrates at a later time through process of cellular respiration to break down and release ATP. So it's one big cycle. So brief look at ATP. ATP is adenosine triphosphate. So you know adenine, the nitrogenous base, along with ribose, along with phosphate, is what you see as a nucleotide. But when you add an additional phosphate group to it, you can hold a lot of energy in this bond here, and you can add one more phosphate to it, and in that time that way it is adenosine with three phosphates, so triphosphate, and once again a lot of energy is contained in this bond. So enzymatically, if you break this down, and remember there are two enzymes, the enzyme that helps build ATP from a ADP molecule, a diphosphate molecule, is an ATP synthase because it is synthesizing ATP. Opposite to that is ATPase, which breaks down the chemical bonds here to release energy. So here we are seeing an ATP molecule breaking down the last phosphate and releasing energy in the process. And that energy can be used to power other cellular reactions. Now that one phosphate is gone, you end up with ADP. So that's the cycle, the ADP ATP cycle. So before we proceed further, we need to understand oxidation and reduction in the context of these biological reactions. Oxidation is simply loss of electrons, and reduction is a gain of electrons. Electrons have a lot of energy in them. So when you reduce a compound by adding an electron to it, it has now gained energy. And here's an interesting thing to note. When a molecule in a cell picks up an electron, it is being reduced, right? When it picks up an electron, it also often picks up a proton at the same time. And you have plenty of protons in available through water, breakdown of water. And those molecules we are going to see, including NADPH in photosynthesis and in cellular respiration, that is NADH. And these molecules, NADPH and NADH, they are molecules that are now containing a lot of potential energy held in the because they are holding electrons within them, high energy electrons in them. Okay, so that's something we need to remember, that oxidation is loss of electrons and reduction is gain of electrons. You can remember it using the acronym OIL RIG. OIL, oxidation is loss, RIG, reduction is gain. And usually, oxidation and reduction reactions are paired with each other. So, oxidizing one chemical into another is supporting the reduction of one into the other. Because they are happening together, they are called as redox reaction. Reduction, oxidation, reduction reactions. Alright, with that background, onward to photosynthesis. Photosynthesis happens in the leaves, and here's a quick look at the structure of leaves. If you take a cross section of a leaf, you can see that on the top of the leaf there is a light waxy coating. Depending on the type of plant, the waxy coating might be thicker or thinner. Under that you have a layer of epidermal cells, and under that there is a column of mesophyll cells, palisade mesophylls. And of course your plants, your leaves need nutrients, and they need water, and the nutrients that they produce need to be transported out. Therefore you have xylem and phloem. These are tubes that transport water. Xylem transports water, phloem transports nutrients. And then there are cells called bundle sheath cells that surround the xylem and the phloem. They're kind of right in the middle of the leaf. And then you have the spongy mesophyll cells, which are kind of loosely arranged, and you can see all of the space between those different leaves. That's where a lot of air is present. And if you take a cross section or a close-up look of what's happening inside the leaf, you will see that, let me show you a quick picture here. You'll see that inside those leaves you have all of these tiny organelles, which are chloroplasts. There are about 30 to 40 chloroplasts in every cell, in every plant cell. Now, before we start looking at the structure of chloroplasts, we also want to note that at the bottom side of the leaf, you have these holes, these tiny holes called stoma. And the stoma is the place where gas exchange happens. You have oxygen and carbon dioxide going in and out, and water vapor also going in and out through those little holes. And these nice cells that are, these differently shaped cells that are covering the stoma, they are called as guard cells. They open and close, and they regulate how much of gases flow in and out. Okay, now let's take a closer look at the inside of a cell. Like I said, inside the cell there are many plastids, chloroplasts. And the chloroplasts, we have seen this before in the context of endosymbiosis, where we have an organism, a smaller bacteria that was swallowed by larger bacteria and eventually became a part of the cell. So here we have a chloroplast that has two membranes, and the outer membrane and the inner membrane. Inside the inner membrane there is a fluid matrix called as the stroma, and inside the stroma you have these flattened discs that are all stacked on top of each other. Those flattened discs are called as thylakoids. And the thylakoid stack, a stack of thylakoids, is called as a granum. And these grana are all kind of attached to each other, and multiple stacks of granum are called as grana. So that's a few things to note. But what you would see here is that a lot of the magic of photosynthesis happens in the thylakoid membrane. So these little flattened unassuming sacs is where the majority of the first step of photosynthesis is going to happen. Photosynthesis happens in two stages. The first one depends on light. The second one, it doesn't need light, so it's called as a light independent reaction. There was a misnomer earlier. They used to call it as the second step is they used to call it as the dark reaction, but that's not true. It doesn't happen in the dark. It just doesn't require light. So the light dependent reaction produces two molecules or two kinds of molecules, ATP and NADPH. And the energy of ATP and the energy that's contained in NADPH are used to run the second step, which is the light independent reaction, otherwise called as Calvin cycle. And the products of Calvin cycle are NADP plus and ADP, which is the source material for making more ATP and NADPH. So once again, it's a beautifully balanced system. Both light dependent reactions and Calvin cycle are dependent on each other because of how they cycle the ATP, ADP, and NADPH and NAD+. But in the process, carbon dioxide from outside is taken in and water is broken down to release oxygen, and the carbon dioxide from the atmosphere that is taken into the Calvin cycle becomes glucose, and we have a perfectly balanced system. So first step, light dependent reaction. Energy of sun comes in the form of photons, which are packets of energy. And the first miracle of life starts here in the thylakoid membrane. Once again, if you need to take a look at this is the thylakoid and the flattened sac that is present inside the chloroplast. And in this membrane is where you have all of this first step, this light dependent reaction happening. Remember that a lot of proteins are embedded in the membranes. That's true of thylakoid membranes as well. Here you have a big complex of proteins along with molecules called pigments present. And there are two types of them. One of them is called photosystem 2. It's a complex of proteins and molecules called pigments. The other one is called photosystem 1, which is very similar to photosystem 2. And in addition, you have these other proteins called cytochrome complexes that are present here. And you have molecules that carry the energy, which you are going to see from one complex to the other complex through cytochromes. So as a first step, energy of photon, the photon comes and hits the pigment molecule. Now, plants have several types of pigments. They have two major types of pigments called chlorophyll A and chlorophyll B. Pigments are very special molecules because they have the ability to get excited by absorbing energy. And different pigments have different wavelengths of light that they can absorb. Remember, light, what we see as sunlight, is actually a mixture of different wavelengths of light. And these pigments, for example, chlorophyll A, is able to absorb a lot of colors, specifically colors that are orange and blue. But one color that they cannot absorb is green. So they reflect the green back. Whatever color is reflected is what we see as the color of that object. So because green light is not absorbed by chlorophyll A or chlorophyll B, we see the leaf as being green because we have all these chloroplasts reflecting all these thylakoid membranes with their chlorophyll molecules. They are all reflecting the green light back from the sun while absorbing other wavelengths of light. So, when a pigment molecule like chlorophyll absorbs the energy of the sun, the electron goes to a higher level. Okay, it just goes to a higher orbit in its energized form, and that's not a sustainable system for the electron. So this process of absorbing sun energy and then especially from light, absorbing energy from light and then going to a higher energy level is called as photoexcitation. It's excited, it's gone from a lower energy level to a higher energy level, but it's not sustainable. So it releases that energy, and that energy is absorbed by the next chlorophyll molecule. And once again, that energy, the electron in that molecule, in that pigment, is going to rise to a higher energy level like this. And once again, it's going to lose that energy so that the electron can come back to its normal state. And that energy that is now released is absorbed by the next pigment molecule, which then has its own electron jump up to the next state. So it's simply, think of it as kind of people in a stadium waving, right? Doing that wave. So it's like that. You take the energy from one pigment molecule and that excites the next one, and that releases the energy, and that excites the next one, and that goes on and on for a while until it comes to the center. That center is called as the reaction center. In the reaction center, you have chlorophyll molecules. It's actually two chlorophyll molecules. And these two chlorophyll molecules, instead of just coming down to normal state and releasing that energy, what they do is they release their electrons. Okay? Until this time, what has been happening is energy has been released, electrons going to a higher energy state and then coming down. Higher energy state, coming down. Here in the reaction center, the electrons that were raised to a higher energy state are just given out. And the energy that is then the electron that is given out, which is having all of that energy from that sun, is now absorbed by a carrier molecule called pheophytin. And this carrier molecule carries that energy to another complex called plastoquinone. You don't need to remember these names, but it's going to be carried on to a complex called cytochrome C. Cytochrome B and F. Cytochrome BF. Sorry, B6 and F. So cytochrome molecule. Just remember this as the cytochrome complex. And from there, it is then carried, the energy is going to be carried into the next complex called photosystem 1. Now, while all of this is happening, whatever happened with your photosystem 2 to begin with is also happening in photosystem 1. Because here again you have chlorophyll molecules. Energy of the sunlight was absorbed by chlorophyll molecules here, and then once again excitation happened, and then back to ground state, excitation, back to ground state, and that keeps going until the final until this stage where it comes to the reaction center. And there, this reaction center is going to give out the high energy electron, which is then accepted by a molecule. That molecule is then going to give out the energy, give that electron to a molecule called ferredoxin. The ferredoxin is then um, going to transfer that to another enzyme called NADP reductase. And what is happening is, in that process, the energy of the sun in photosystem 1 is now transferred in the form of electrons to NADH reductase, NADP reductase, I'm sorry. And what it does is, it takes a molecule called NADP plus. NADP plus absorbs an electron, and if you remember what we mentioned earlier, when it picks up an electron, it also often picks up a proton, because protons are present everywhere. What it does is, NADP molecule, as it absorbs the electron and it becomes energized, also takes a H plus, a proton, and becomes a high energy molecule called NADPH. Okay. Remember, all of this energy that made NADPH came directly from photosystem 1. Now, in this reaction center in photosystem 1, because it gave up that electron, it is now short of an electron. There is an electron hole here in this reaction center. And remember the electron that came from photosystem 1 that jumped from here to the carrier molecule and then to cytochrome C, and then it was carried over here. That electron is accepted by our friends here in this reaction center. In that process, this reaction center of photosystem 1 is happy because its electron hole is filled. Okay. So we have accounted for the electron that came from the absorption of photon from here. We have accounted for the hole that was left behind in photosystem 1 at the reaction center because of the electron that it received from photosystem 2. But what is not explained yet is what happens to the electron from in the electron loss in photosystem 2. You remember that here we lost the electron to this carrier, phaeophytin, which means that there is an electron hole here, right? How is that going to be filled? For that, we need water. Now, water comes to the plant leaf from the water that it absorbs to the root. Water is H2O, 2 H's and 1 O. So the H and the O are broken down. And now you have a lot of protons here, H plus. Remember that a proton is simply, so when you add a proton and an electron together, you get a H, a hydrogen atom. A hydrogen atom is simply a proton and an electron together. So now you have water being broken down into 2 protons and an O minus, half of an oxygen, right? H2O, because O, half of an O2. So we have that O here. And this, during this breakdown, the electron that is released is going to be taken in by this reaction center, the reaction center of photosystem 2. So that way, photosystem 2 is also happy. So what we have ended up here is a whole bunch of protons because of the breakdown of water. The electrons that were created were used because remember H is equal to H plus and E. So the electron there is the one that has gone here. So you have protons left behind, but then you have oxygen. And oxygen is going to come together, half of O2 plus another half of O2 is going to give O2, that is oxygen. Oxygen is a gas. and that's easily diffusible through membranes. So it diffuses through the thylakoid membranes, it diffuses out of the chloroplast, it diffuses out of the plant cell wall, uh, and then it gets into this little space here and in these spaces, and then it is sent out through the stoma. Okay, so all of this is um, carefully coordinated, but a couple of things are still hanging loose. One is the idea that you have created a lot of protons on this side. Okay, so you have the thylakoid membrane, and we have between the thylakoids you have the thylakoid space, this tiny little space here. This is where you're accumulating a lot of the protons, which means you have a lot of charge, a positive charge on one side, and outside of the thylakoid membrane, that's a stroma, you don't have as many H plus molecules, H plus ions. So now you have created a gradient. And the cell uses this gradient to generate energy using this powerful molecule called ATP synthase. So let's take a look at this membrane again. Let me see if I have a different picture here. Yeah. So here's your thylakoid membrane, and this is your thylakoid space. As the electrons from the breakdown of water are going in, and you're ending up with a lot of H plus, you're creating a gradient. Now, we also mentioned that the electron is going to be passed through a complex called cytochrome C before it goes to photosystem 1. Cytochrome C, when it accepts the electron, it becomes charged, and in that process what it does is whatever protons are already existing in the stomata, in the stroma, yeah, see, whatever protons are already present in the stroma, it moves it against the concentration gradient and into the thylakoid space. Okay, so take a second to think about that. There's already some protons here, and it is going to move it against the concentration gradient just because it got an electron when it was passing through it from photosystem 2 to photosystem 1, and that opened up that force that became acted as a pump and then pushed a lot of protons here. So essentially, between those two reactions, between the breakdown of water and between the cytochrome complex becoming activated by the electron that it absorbs, you're ending up with a proton gradient. And that proton gradient activates a molecule called ATP synthase, which is bound to the membrane as well, to the thylakoid membrane as well. And ATP synthase is this powerful molecule which is magnificent. What it does is it responds to the change in gradient, to the gradient difference of protons, and opens up and allows the protons to pass through it very fast. As it moves through it, it is mechanically designed to act as a rotor, and it's a very efficient rotor. And as that rotor spins in the space here, in the stromal space, it's going to release ATP. Okay, so here in this case we are looking at a mitochondrial membrane. You're going to see the same design in the thylakoid membrane as well. It's the exact same thing, ATP synthase. So what ATP synthase is done is, as the protons move through it in the outside of thylakoid space, that is in the stroma, ADP becomes converted into ATP. So what has happened is you have NADPH that was created because of the movement of energy through photosystem 1 and receiving by ferredoxin and then NADPH, NADP plus reductase. You ended up creating NADPH from NADP plus, and then you have ATP that was generated because of the movement of the protons from inside the thylakoid membrane to the outside. So now the ATP and NADPH, which are the products of the light dependent reaction, are going to power the next step, which is the light independent reaction or the Calvin cycle. Okay. Alright, now we go into stage 2, which is the Calvin cycle or the light independent reaction. Calvin cycle is where carbon dioxide from it in its inorganic form becomes converted into an organic molecule like glucose. And to do that, it is going to use the energy of ATP and NADPH that was generated during the first part of this photosynthesis pathway. So the starting point of this is a 5-carbon molecule called ribulose 1,5-bisphosphate, also called as RUBP. 5-carbon molecule. This 5-carbon molecule joins with carbon dioxide. And where does that carbon dioxide come from? Carbon dioxide comes from the air because it enters through the stoma and into the cell and into the chloroplast. So all of this reaction is happening in the stroma of the chloroplast. So the ribulose 1,5-bisphosphate joins with carbon dioxide from the atmosphere, and this beautiful magic molecule called Rubisco does the most extraordinary thing. It takes inorganic carbon dioxide and joins it to ribulose 1,5-bisphosphate and makes a short-lived 6-carbon molecule. Rubisco is the most common protein in the world. There are, because every plant that does photosynthesis depends upon Rubisco for doing this critical step. Now, Rubisco, once it creates that 6-carbon molecule, that 6-carbon molecule gets broken down into 2 3-carbon molecules. Those molecules are called as PGA, phosphoglycerate, and 3-phosphoglycerate. So this 6-carbon, so the 3-carbon molecule phosphoglycerate, in a couple of steps, becomes converted into a molecule called G3P, glyceraldehyde 3-phosphate. And glyceraldehyde 3-phosphate goes through a series of steps, and then some of it becomes converted into ribulose 5-bisphosphate, and some of it becomes converted into glucose. So, just to recap, carbon dioxide from the atmosphere joins with the pre-existing molecule called RUBP, and in a process called carbon fixation using an enzyme called Rubisco, becomes converted into a short-lived 6-carbon molecule, and that 6-carbon molecule breaks down into two molecules of PGA, and that PGA molecules becomes glyceraldehyde 3-phosphate or G3P molecules. So essentially the step is the reduction of PGA to G3P. Now G3P, some of it is going to regenerate RUBP and some of it is going to be used to make glucose and other sugars. Now let's take a closer look. Let's take a molecule of glucose. Glucose has 6 carbons. Okay, so if all of the molecules, of all of the atoms of all of the 6 carbon atoms of glucose have to come from carbon dioxide, we need 6 molecules of carbon dioxide, right? I'll repeat that. If all of the 6 carbon atoms that make up the 6 carbon glucose molecule have to come from carbon dioxide, then we need 6 carbon dioxide molecules. So we need 6 carbon dioxide molecules coming in, which means we need 6 RuBP molecules to interact with them. So 6 RuBP molecules plus 6 carbon dioxide molecules gives 6 of the short-lived molecules, the short-lived intermediates. And because now that's breaking into 2, each 6 carbon molecule is breaking into 2, we are going to end up with 12 PGAs, and the 12 PGAs are reduced to 12 G3Ps, and we have 2 G3P molecules dedicated to make one glucose molecule. Okay, so 2 G3P molecules is 6 carbons, and the remaining 10 G3P molecules are going to be converted into 5 molecules of RuBP. I'm sorry, 6 molecules of RuBP, because RuBP is 5 carbons, so we're going to make 6 molecules of RuBP. So if you look at one of the exercises that you can do is pause the video here and track how many carbons are there. So it has to tally to a total of 30 here and 6 coming from the outside. And so if you started out with 36 carbons with the short-lived molecule, at the end of the day you should be able to account for 36 carbons. So just pause this video if you need to and take a look at how all of these things are balanced. But what you need to see is how the ATP, the energy of ATP, is used to drive different parts of the Calvin cycle. So you have 12 ATP molecules being broken down into 12 ADP molecules in this step, 12 NADPH molecules being converted into NADP plus molecules and phosphate in this step, and then have 6 ATP molecules are broken down to 6 ADP molecules and 6 phosphate molecules in this step. So all of these essentially mean that ATP and NADPH are powering the Calvin cycle. And what are the end products of this conversion? ATP becomes ADP, and NADPH becomes NADP plus, and again here ATP becomes ADP. So let's step back and look at the bigger picture of things. For this reaction to run, for the light dependent reaction to run, we need NADP plus and ADP, and we just saw where that is coming from the Calvin cycle. And then the product of light dependent reaction is ATP and NADPH, and those are the things that are running the Calvin cycle. And we saw how carbon dioxide is entering here in the Calvin cycle step, and how in the first step the breakdown of water is releasing oxygen. And so very quickly what we have done is we have taken energy of sun, we have maintained a very balanced system inside. We took water and we broke it into oxygen. We took carbon dioxide from the outside like this and then made it into glucose, and we still have a fully functional system. And these byproducts are what we depend upon for our survival, glucose and oxygen. And plants need glucose and oxygen as well, because they also need the energy that they have stored in the form of carbon-hydrogen bonds in glucose. So it's a neatly balanced system. Cellular respiration happening in the mitochondria, photosynthesis happening in the chloroplasts drive each other within plants, and in animals we don't have photosynthesis, but we are able to take we are actually able to steal, either directly or indirectly, the energy that is captured from the sun through the chloroplasts into the form of sugars, and we eat them, we consume them, and we have enzymes that can break them up, and the enzymes that we use to break them up in the mitochondria release energy in the form of ATP molecules, and then the energy that is used for catabolic processes and anabolic processes. Actually, not catabolic, but anabolic processes, building up stuff in metabolism. So, here's an interesting thing to look at. So, you might wonder why we have multiple types of chlorophylls. So, we saw that chlorophyll photosystem 2 and photosystem 1 have different types of chlorophyll molecules. Both are pigments, both can absorb, both are reflecting green, but there is a difference in the wavelengths of light that they are absorbing. Chlorophyll A, which is the dominant molecule, absorbs a lot of orange, and it also absorbs a lot of blue. Chlorophyll B absorbs more of the violet range of material, but also a little bit of orange. And you have other molecules like phycoerythrin and carotenoids, which are supporting molecules. They can absorb energy at a different wavelength. They can absorb green, for example, and then they can transfer that energy to chlorophyll A and chlorophyll B. But why do we have to have so much redundancy? Right? Why do we have so many different things? So that's because of a concept called fitness. It's not physical fitness we're talking about. We're talking about molecular fitness, which actually leads to ecological fitness. So here in this case, we have molecules, we have a system where we are able to absorb different wavelengths of light. So even if you have a lack of availability of one wavelength of light, you still have other molecules that can capture the other molecules of light. Or in other words, it makes it more efficient. If you have multiple wavelengths of light that you can capture by different molecules, then you are able to survive in different environments with more efficiency. So that is one of the concepts of fitness, which is to say that variation in the number and types of molecules within cells provide organisms a greater ability to survive and or reproduce in different environments. So just take a second to observe that. If you have more variety of molecules, or in other words more variation in the number of molecules and types of molecules, you have a greater ability to survive and reproduce in different environments, which also explains how organisms are able to, from an ecological standpoint and from an evolutionary standpoint, adapt to different conditions. And again, that is also called as fitness, evolutionary fitness. Another example, another variation of this fitness is where you have you have variation at the molecular level, where you have variation at the molecular level, which provides the organisms the ability to respond to a variety of environmental stimuli. An example of this is when we are looking at your plasma membrane. Remember how we saw that we have saturated and unsaturated tails? So because we have a variety of different um, molecules in the tails that are making up the tails, depending on whether there is heat in the environment or cold in the environment, and because saturated and unsaturated fats have different ability to impact the fluidity of the membrane, you are able to adapt to cold and heat by because of the variation in the level in the types of molecules that are making up the plasma membrane. So again, that is an example of fitness. So fitness is discussed as a separate um, chapter, but since we are covering it here, we might not need to make a separate chapter out of it. But that's what fitness is. Fitness is what happens at and in this chapter at least, or in this context at least, it's what happening at the molecular level that will give it the ability to respond and survive in different environments. Okay, so now we're coming to the concept of C3, C4, and CAM plants. Very simply, this is plants adapting to different environments, giving them an opportunity and an ability to survive in the conditions of heat, for example. So the majority of plants, about 85% of plants that include rice and barley and wheat, all of these guys follow the very same pathway that we just discussed, where it is making a molecule called G3P, glyceraldehyde 3-phosphate. So G3P as a molecule, because G3P is a 3-carbon molecule, the plants that utilize this one-step process are called as C3 plants. C is for carbon, so 3-carbon plants. But plants like maize and sugarcane that grow in hot conditions, it's about 3% of plants, they use a different strategy. Here's why. For this, we need to go back and look at the structure of the plant leaf. When the stoma opens and you have carbon dioxide going in and oxygen also being present, and you have these mesophyll cells where most of the in C3 plants at least, most of the photosynthesis reactions are happening either in these cells or in these cells. Okay, the spongy mesophyll cells or the palisade mesophyll cells. So here in C3 plants, first let's start with C3 plants. So what happens in C3 plants is that there is a process called photorespiration. Photorespiration happens when the conditions are hot, let's say when it's really hot, and the stoma closes, trapping carbon dioxide as well as oxygen. And what happens there is that the oxygen is used by Rubisco. Rubisco doesn't differentiate very well between carbon dioxide and oxygen. So what Rubisco does is, instead of just using carbon dioxide to run this reaction, sometimes it can use oxygen that is trapped here to run a reaction called photorespiration. I don't think I have a picture of photorespiration here, but photorespiration. So photorespiration, what happens is that you lose about 25% of the carbon dioxide in a cycle that is not productive for the plant. It is not going to make G3P and therefore carbohydrates. It's a wasted cycle. So 25% of carbon dioxide is lost under hot conditions on hot days. So that is a disadvantage for the C3 plant. So plants that live in hot conditions, what they have learned to do is, as soon as they absorb the carbon dioxide in the mesophyll cells, which are closer to the outside, the spongy and the palisade mesophyll cells, what it does is it takes in the carbon dioxide and converts it into malic acid or malate, which is a 4-carbon molecule. And that 4-carbon molecule gets transported to these cells that are surrounding the xylem and phloem. They're called the bundle sheath cells. So they are inside and they don't have much oxygen in them. That's the trick. They don't have too much of oxygen here. So the malic acid or the malate that is created is now going to get into the bundle sheath cells, and there it gets converted back into carbon dioxide. And that carbon dioxide inside the bundle sheath cells now enters the Calvin cycle without the presence of too much of oxygen. So it's a very efficient process. So under hot conditions, under hot days, your plant can take in carbon dioxide and oxygen and close very quickly. And the oxygen actually that is generated from the first step of the photosynthetic reaction, which is the light dependent reaction, that is also used. It doesn't have to take oxygen from outside sometimes. It can just use the oxygen from the first step of the reaction. And that oxygen is not going to be reaching the bundle sheath cells, so you are kind of insulated from photorespiration here. But what about plants that are growing in extremely hot conditions like deserts? Now, this strategy is going to work for them. The C4 strategy is not going to work for them. CAM stands for Crassulacean acid metabolism, which is basically the group of plants, the Crassulaceae, in which they were identified. Carbon dioxide in getting into the plants happen during the night time. So during the daytime when it is hot, the stoma remains closed so that it doesn't lose any water vapor escaping from it. So during the night time when temperatures are colder, it can safely open the stomata without the fear of losing water. So carbon dioxide that enters inside during the night time is converted into malic acid, malate, and then converted into carbon dioxide. And during the daytime, that carbon dioxide enters the Calvin cycle. And there, because during the daytime you're getting your sunlight energy and you're making your ATP and your NADPH from your first step, the light dependent reaction, at that point the carbon dioxide that they obtained in the night is used to run the Calvin cycle and the ATP and the NADPH from the light dependent reaction. So that way there is no fear of losing water in this step, and the plants can survive the intense heat while taking advantage of the heavy sun. And in all of those cases, you are able to generate glucose efficiently. So here's a comparison. Temperate plants for C3, which is 85% of them. C4 plants, which use hot weather, where they are basically separating the fixation and the Calvin cycle by using bundle sheath cells and mesophyll cells for 2 different steps. And then we have CAM plants, which are used, the CAM strategy, which is used by desert plants by separating it night and day. Okay. So that's it. Hopefully at this point you're comfortable with the energy cycle, which is how energy from the sun is moved in the form of small molecules, NADH and NADPH and ATP and all these energy molecules are now fueled by breaking down to run other biochemical reactions. We also saw what reduction versus oxidation is, and we also looked at the 2 different stages of photosynthesis in detail. We looked at how light dependent reactions for photosystem 2 and photosystem 1 are taking in energy of the sun, using pigments to absorb the photons get excited, and you have reaction center where the electrons are actually kicked out, and those electrons are now, the electron holes are now filled in by either breaking down of water in photosystem 2, or the electrons from photosystem 2 that are used in photosystem 1. And in the process we are seeing a formation of H plus a proton gradient, and then the gradient is going to activate ATP synthase, which is going to move protons, and in the process create a mechanical system for converting ADP into ATP. Whereas we also saw that in the last step, the photosystem 1 is going to use the last electron that it is sending out is used for creation of NADPH. And we saw how the NADPH and the ATP that were created are then used in Calvin cycle, using Rubisco as an enzyme to take carbon dioxide from outside and RUBP that is already present in the system to go through a cycle where RUBP is regenerated and a little bit of glucose is created every time. And we also saw that this idea of fitness, very briefly, where we have variations of molecules and variations of types of molecules that will help that will help a plant survive, or to help organisms survive different conditions, different environmental challenges. And as a part of discussing adaptations to environmental challenges, we saw that C3 plants, the ones that we discussed before, also have variations called C4, which are separating out the two types of cells, bundle sheath cells and mesophyll cells, and also night and day reactions, as we saw in desert plants called as camp plants. That in essence is photosynthesis. Hopefully it all makes sense. And in the next session we are going to study how the energy that is now caught or captured in the glucose molecule is now going to be broken down as needed in the mitochondria in both plants and animal cells as a part of what we call as cellular respiration. Okay. Hopefully this was helpful. Thank you.